Good evening, everyone, and welcome to tonight's Kraw Lecture. I'm George Kraw. The Kraw Lecture series features acclaimed UC Santa Cruz scientists and technologists who are grappling with some of the biggest questions of our time. Sara Abrahamsson, an assistant professor of electrical and computer engineering here at UC Santa Cruz, is at the forefront of innovations in optical microscopy. She invented a technique called aberration-corrected multifocus microscopy, or MFM, which enables three-dimensional imaging of living cells. More recently, she showed that MFM can be combined with another technique called structural illumination microscopy, SIM, that provides super resolution beyond the classical limits of light microscopes. Now, Abrahamsson is working with UCSC biologists to build and test the first multifocus structured illumination microscope. Tonight we learn how Saralab here at UCSC invents and custom builds new types of optical elements and imaging systems to support these biologists and others. Before we begin, I would like to share a few details about the event tonight. We are using a webinar tool, so there is no chat function. We will have an opportunity to answer your questions at the end of the program, and we invite you to submit your questions into the Q&A box at any time. Tonight's event will be recorded. Our conversation tonight will be moderated by Alexander L. Wolf, who has served as Dean of the Baskin School of Engineering and Distinguished Professor of Computer Science and Engineering at UC Santa Cruz since 2016. Professor Wolf received MS and PhD degrees in computer science at the University of Massachusetts Amherst before joining AT&T Bell Laboratories as a member of the technical staff. He was a professor of computer science at the University of Colorado Boulder for 14 years, where he held the Charles V. Shelkey Endowed Chair. Wolf spent another decade as a chaired professor at Imperial College in London, prior to which he co-founded and taught at the Faculty of Informatics at the Università della Svizzera Italiana in Lugano, Switzerland. He has also served as a distinguished visiting professor at the School of Software at Tsinghua University in Beijing and a visiting professor at the Università di Roma, La Sapienza. Wolf's technical interests span the areas of attributed systems, networking, and software engineering, and he has served as a consultant and advisor to universities, tech companies, and a number of science and technology institutions in Europe. He is a fellow and former president of the Association for Computer Machinery, ACM, and a fellow of the IEE and the British Computer Society. Professor Wolf currently serves as a member of the Computer Research Association Board of Directors and the American Association of Engineering Education Public Policy Committee. Welcome, Dean Wolf.
Thank you, George. I'm very happy to be here and to introduce this talk. Thank you all for attending, and I'm very excited to join you in learning even more about the work that Sara is doing. Sara and the students in her lab are inventing new types of optical elements and imaging systems, and these technologies are enabling scientists to study living cells in unprecedented detail, and in particular in 3 dimensions and in real time. They design and they fabricate their own diffractive optics and use nanofabrication for simultaneous 3D imaging. So the work here that we're going to be seeing is truly, hearing about is truly revolutionary and making great advances and helping scientists, in particular biologists, learn more about the processes underlying underlying cell mechanisms. Sara came to UC Santa Cruz because of the strength of our campus in bioelectronics engineering and biological sciences. Sara's work contributes to the UCSC Life Sciences Microscopy Center, which provides researchers at UCSC with personalized assistance on various aspects of imaging, from experimental design to training on the shared microscopes and imaging analysis or image analysis. Her expertise is really in demand across the campus and well beyond. The Saralab is a special place, and I've had the opportunity to visit it, and I would hope you would get that chance too. The lab involves all of undergraduates, master's students, and PhD students working together to build and test out these technologies. The first undergraduate to graduate from Sara's lab, Brandon Lynch, received the Chancellor's Award for Research Excellence and Creativity in his undergraduate studies. Some of you may have attended a recent Kraw Lecture that was presented by Holger Schmidt. He also works at this exciting intersection of electronics, optics, and biology. And as I said, this is really revolutionizing how we are understanding biological processes for health and disease. In this evening's Kraw Lecture, we're really going to be getting a little peek at why we are entering a golden age of microscopy for biologists. The first microscopes, of course, were optical, but very primitive by comparison. These new techniques that Sara is inventing, as Sara and her students are inventing, are leaps and bounds ahead of where we were just a few years ago. So please join me in thanking Sara, who is a professor in the Baskin Engineering's Electrical and Computer Engineering Department. And Sara, I will hand it off to you. Oh, let me just say, please enter your questions in as we go. I may choose to interrupt Sara with a question, and we're also going to break occasionally to take some of your questions. Sara.
Good. I will share my screen with you here, and let me get it up in full screen. Can everyone see that good? I hope so. I'm super glad to be here telling you about our wonderful lab and the research we are doing. My real passion is optical systems and things that glow in the dark. And what better thing to do than optical systems design? You get to design beautiful things and you get to build beautiful things and then try to commission them and interact with biologists to do a fantastic work that, you know, is so difficult. I could never have studied biology. I studied physics. It's really marvelous how these fields all come together. If there's one thing that I'm even more happy about, it is that this is back in school. So this week we are returning to class, we are returning to lab. I've spent the last couple of days this week with my students in the laboratory, and we're so happy to be together again and to be able to work and work together and interact and really make progress in these exciting inventions that have really suffered from the lockdown, and we're just so glad to be back. So we design optical systems with the goal of visualizing live cells and organisms. And I'm giving you a little example here of a cell, and we'll talk maybe a little bit more about that. But what we're looking at is single molecules of RNA polymerase diffusing through the cell nucleus. And we're imaging this live in 3D, which is something really spectacular. Like Alex was saying, we've come a long way from the original microscopes where you actually had to draw what you saw in the eyepiece because there were no cameras yet, right? So optical technology is actually really, really old. Some of my students here in the slide are busy in the lab building, busy in the Nanofab making diffractive optics devices. And that's pretty much a big part of what we do here. So here's an example of a cell. And I just wanted to flash that up here to point out that this is truly amazing work by one of the other postdocs in my PhD lab back in the days. And even from that, we've come a far way. We are looking here at mitochondria, and depth is encoded with color. So this is a 3D image of this living cell. And if you look closely, you see inside these mitochondria little structures. So for a long time, we've been able to see cells. We've been able to see even the organelles inside the cells, like the nucleus and the mitochondria. But today, with the Superresolution techniques that received the Nobel Prize in chemistry in 2014, we actually can go beyond the resolution of the diffraction limit, and we can see the structures inside the structures of the cell. This is quite mind-blowing, I have to say, and it doesn't cease to amaze me. As I'm sure you're aware, cells don't really glow in the dark in general. So this is labeled cells, and they're labeled with these fluorescent proteins that glow in the dark, and they're actually genetically expressed so that we could look at living organisms. And you can label particular proteins, and you can label the different proteins in different colors and see how they interact with each other. So let's start on optics. This will be an optics lecture. I'm an optics person, and there's not much to do to stop me to talk about that. And I'll start with photography, since I think that's the most intuitive part of optics, since many of you probably have used a real camera, although a lot of people today, including myself, take a lot of pictures with their cell phone. But the photographic camera is really cool, and it's this aperture that it has that we can set and increase and decrease, which is called F number in photography. I'm sure you're all aware of the wonderful group of photographers in California who call themselves the F64 group. So Ansel Adams and the whole team of men and women photographers who wanted to take 2D images with extreme depth of focus. So this is a 3D scene that we're looking at, but everything is in focus because they've closed down the aperture to get this extreme focus depth. So that's one way of imaging a three-dimensional world. The opposite way of imaging this three-dimensional world is to open the aperture as much as we can, because what we can do then is we can slice this world up. So this 3D scene, all of a sudden, we can choose just one plane of it to image. And the reason I chose this specimen, it's a scene, I guess. In biology, we always talk about specimens, is that I was a Leon Levy Fellow at the Rockefeller University, and I was supported by Ms. Shelby White through a Leon Levy Fellowship, and she also supported the Brooklyn Botanical Gardens. So it was really great to be able to integrate those two things into demonstrating to you depth of focus and how we can use this depth of focus and reduce it to get a 3D view of the world. So I've imaged a little cherry blossom here in macro setting. So we're moving towards microscopy here, you can see. And you can see that with this big aperture, we can slice out everything but one focal plane. And this can give us a 3D rendering where we know where things are in 3D, moving towards being able to visualize 3D space. And another thing that I just wanted to get everyone to think about is temporal resolution. So how fast we need to image to capture something. This is an image by my PhD student, Eduardo Hirata from Peru. And we can see here that if you have a long exposure time, maybe you get a really nice, beautiful view of the water and this waterfall falling. But if you want to catch the droplets and stop time, you have to have a very short exposure. And this today, we have cameras that move really fast. Back in the days, Harold Edgerton, one of my favorite photographers, he used a mechanical shutter and film. So we couldn't really image this fast. And then he invented this trick called flash strobe photography, or possibly someone else may have invented that before him. Don't quote me on this one. He was one of the forefront people in designing these. So this is a photograph from the Wimbledon tennis tournament. And we can really see how now we're getting information out of this moving scene. We can see how fast the ball is going if we know how fast we flashed the strobe recording this image. And we can see the shape of the serve and where everything is going. And of course, today we have electronic cameras, both the CMOS and CCD sensors that can run extremely fast. CMOS cameras can run maybe 1,000 frames per second if you have enough. If you have enough light to have such a short exposure time. So we really come a long way with that technology, and 2D imaging is really fast. And the issue here now, we want to take a 3D image, and that is we need to scan through focus. So scanning through focus like this, sure, it can be really fast. We can have a piezoelectric control stage that runs through the sample back and forth and records the 3D focal stack. But it's always going to take some time. So we can appreciate that the temporal resolution here, this is a classic problem in optics, in wide field imaging, in photography, in microscopy, anytime where you have a system that forms a 2D image, but what you actually want is to capture the 3D world, like multiple cells in a culture, multiple neurons in a brain, or whatever your biological specimen of interest could be, or maybe it's a scene outdoors. So this was, so here we go on to microscopy. And the microscope is really a lot like the camera. It just has so much more magnification. It has some little differences in the optics, but basically we're still forming this 2D wide field image. So if we take one image on our electronic camera, we get a 2D image, and to get a 3D image, we record this focal stack, and we can form a 3D view of our specimen. So when I started my PhD in San Francisco, coming there from Sweden as a young engineering physics master student, one of the professors there grabbed me and told me, I want you to do this thing. I want you to make 3D instantaneous imaging. And what you're going to do is you're going to find the way, using whatever means you like, to capture this focal stack in one exposure, just one exposure. And you know, can we do that without losing resolution was the question, because there were a few methods where people tried to do this, but you always lost resolution. And in microscopy, we want to see the smallest things. We want to see single biomolecules. And the answer to this question, thankfully, was yes, because I did a PhD for many, many years. I'm almost embarrassed to say how many, because it's a little bit over the time limit that you're really supposed to do a PhD on. But it was a wonderful experience, and I will tell you about exactly the optics, how this works. But I wanted to first just stop and say, are there any spontaneous questions on 3D imaging to try to make this a little bit interactive?
Well, Sarah, there are a couple of questions that came through, and I can convey those. When you first showed the image, you showed them with multiple, when you showed the image of the mitochondria, there were multiple colors, and you very quickly said it, but just to be clear, what you were saying was that the multiple colors were used to indicate different levels of.
Yeah, visualizing 3D data is as hard as recording it, I think, unless you're sitting with your, gaming station and your 3D glasses. So I'm afraid I've mixed a few different ways to convey 3D images in this talk. And that image was encoding depth with color.
Okay, so the different colors indicated different depths. And I suppose if you were able to do this in a way where you could wear glasses, 3D glasses. Yeah,
you could see the whole thing.
Then you could really see it in your mind. Another question is, how does this relate to techniques that have been, we've heard about, like PET and CAT scans and MRIs and things like that, or they also seem to provide 3D images, right?
Yeah, so those imaging techniques are tomographic. And the important difference between them and visible light imaging is that with visible light, we can really image 3D samples. So those image formation, it doesn't really come as a 2D image from a lens, but it's multiple exposures taken from different perspectives. So if you imagine that you have this little object here that you want to image, I'm looking around for an object, but imagine an object here. You do a shadow picture and you have two screens like this, and you rotate them all the way around the sample, and then you combine using the Fourier slice theorem or the radon transform that into a 3D view. So it's still a sequential recording. And those techniques are very powerful. They're working with different the parts of the electromagnetic spectrum, and the power of light. Like the reason I use visible light is, we can really see living things with it. If we have a model organism, we can shine light through it, and we can see it when it's living. MRI can also be done in living people. I've had MRIs myself, unfortunately. And then, you're looking at a living sample, but the resolution that you get can never get down to that level of the single molecule inside the cell nucleus going about doing transcription. So those techniques are really cool. And they're a different way of getting a 3D view of the world. And of course, holography is another way to get a 3D view of the world.
Yeah, that was a question about how this relates to holography.
Yeah, there is a really brave lab. I forget where they're at now, but they're trying to do holography in microscopy, and it's absolutely fascinating.
Okay, great. There's also a question about diffractive optics and how that relates to what you're doing.
I'm going to talk a lot
Okay,
about that.
we'll wait then. We'll wait.
Yeah.
All right, why don't we go ahead then?
Yeah. So here's a little teaser of the multifocus microscope, and those are the diffractive optics in that ray tracing diagram, and I will go into the very detail of how those work. I just first wanted to show you this focal stack of a cell image in the multifocus microscope, and you can see if you read like a book in English that starts at the left and the top and goes row-wise downwards. You see the top layer of this nuclear pore complex, which covers the nucleus of the cell. It's the pores that transport biomolecules in and out of the cell nucleus that are labeled here. And here we have on top, I don't know, I cannot see my cursors, so maybe you cannot either, but we can see this egg-shaped little nucleus, and we see all of these nuclear pores fluorescently labeled on the membrane. And using this technique, we can take one snapshot and get these 9 focal planes that form an image, a 3D image of that volume. And I haven't tried to do a 3D rendering of that in this particular frame, but we'll come to that later. So this technique really then enables us to do this type of imaging in 3 dimensions. With very fast cameras, we have this view of the entire 3D space of the cell in the form of 2D slices. And now we get to my favorite topic, diffractive optics. And here we're really harnessing the wave nature of light. So in Santa Cruz, we're very familiar with ocean waves and with ripples on water. And even though when we think about light, we usually think about reflection of light in a mirror as light comes as a ray, or in our eye or in a lens where light is refracted. But diffraction is really the wave nature of light. And light really ripples when it hits an obstacle, just like water does. We normally can't see this because it's a little bit of a monochromatic phenomenon. But in physics class, we've all seen the double slit Young's experiment. And we see that when light comes through this little barrier, it actually doesn't go straight ahead, even if it came in straight, because it interacts with matter and it ripples like water. And when we put multiple slits together, we get these beams to go in different directions, and we get positive and negative or constructive and destructive interference of the light waves, so that the light really goes into different directions and different shapes. And we can control this with diffractive optics. If we design these functions, we can control light almost any way we want. So the first example I showed here was when we had a little barrier that stops the light and then transmits light and stops light and transmits light. But it turns out if you do that, you lose photons. And when we're doing microscopy, we're looking at fluorescent proteins, and we really don't want to lose any photon. Every photon is sacred. You see a single molecule, you do not have many photons to capture. So we use transparent diffractive optics. We have little phase steps in them, little stair steps that introduce a phase shift in the different parts of the light wave that makes the same effect. So those are called phase diffractive gratings. And here's me holding one of these homemade phase gratings. And you can see it's transparent, and we can't even see the pattern because the pattern is so small. Here's an example of a pattern that would generate a response. So I've tried to indicate here with the levels, the little stair steps in this beautiful shape. And they will steer light through this grating. So the grating is transparent and we have little stair steps in the glass that we've etched there with the reactive ion etching. So the simplest grating we can think of is probably the line grating. So if we have this line grating, it's almost like those little slits, and we choose a laser pointer through that grating. We end up with the ray being diffracted in upwards in what we call positive diffractive orders. Some of it goes straight ahead and some of them go downwards. So that's probably the simplest grating we can imagine. What happens if we shoot an image through that? So we shot a laser pointer there, and if we shot that at the wall, we would get a spot on the wall. And now if we shot that through this line grating, we would get multiple spots on the wall. And it turns out if we shoot an image through that, we get multiple images. So that's a start towards this simultaneous recording of that entire focal stack. So using this diffractive optics device, we can multiplex the beam. Now, the line grating shoots images up and down, but we can make this grating shoot images just the way we want it. So in multifocus microscopy, what we wanted to do was we wanted to shoot 9 images that fit in a square camera sensor. And I wrote, and it's probably the worst code you will ever see because I'm not a software developer, my brother is. I wrote it in MATLAB, and I had this target function of these 9 spots. And I used the 2D-dimensional Fourier transform, transforming back and forth to find this pattern. And it turns out that what you get is this little goblet shape. And if you want to try this on your own, you can generate any kind of shape. Like these spots is kind of boring if you just want to make a diffractive optic. You can make a little picture of a guitar, for example. We did that in the Nanofab at Cornell. But here we're making these spots, right? So here's the pixel flipper running, and you can see it's on the left here, we're generating the pattern, and on the right, these are the 9 central diffractive orders, so the positive and negative and 0th diffraction order. And it's really cool, and we can use this to design basically any grading function we want. This is probably the most boring one you'll ever see. It's a 3x3 fan-out array.
We have a couple of questions, if I interrupt you. Let's see. So what's the point of doing simultaneous imaging, having 9 images? Why is that important to have 9 images?
Then I've dropped you very early on. So the idea here was that we would get the 3D image.
Okay.
So the 2D imaging here, which is so fast, when we do the 3D imaging and we scan through the sample, it takes time, right? So we want to capture a single molecule diffusing through the cell nucleus. We're going to have to image as fast as the camera can run. And then if we're taking a 2D view of that little molecule is going to escape us. We're not going to be able to follow it. So now we want to do an image of the cell nucleus itself, an image of some structure inside it, and an image of this biomolecule going through it. So we want to be fast. We want to be fast as we can, and we want to do 3D. So the idea here, again, was that we scan through the focus in classical 3D imaging, but now what we want to do is we want to capture all of these focal planes at the same time. So using this diffractive optic, the first step we do here is we multiplex the image. So again, if I make this grating that multiplexes a spot, if I put an image through it, it multiplexes the image. So I have my microscope, I form an image of this cell nucleus, and now I put this diffractive grating in, and bam, I get multiple images of that sample. So that's step 1 here. So I designed the target function in order to get all of the light into one of the images. Because again, we're looking at sometimes at single molecules. If we split this up into more orders, we're going to lose light. So that's why this design process is so important in diffractive optics in microscopy, that we really want to squeeze out every photon and get the perfect pattern. And this algorithm has now been designed here to give us 9 spots. So if I shot the laser pointer, I would get 9 spots. But now I shoot the image through it and I get 9 images.
Okay.
And in the nanofabrication facility, it turns out we can make all of these gratings, and it's really fun to work in them. So if you design this diffractive optic that you want to make, the United States has this fantastic system of open nanofabrication facilities, not only to academics, but also to companies and individuals, where you would write a proposal and tell them, hey, I invented a diffractive optic, or I invented a new type of camera sensor, and I want to go to the nanofab and I want to make it. They will take you in and they will train you and put you onto the state-of-the-art tools to make these type of devices. And my students and I have had so much fun making stuff in these labs. And you can basically do with diffractive optics anything you want. But as someone pointed out, what's the point of just multiplexing the image? Now we just have the same image 9 times. That was not at all what we wanted, right? So what we wanted was to focus shift. And it turns out that diffractive optics are so powerful because we can control the wavefront completely, that we can use this same grating to do the focus shift. So first of all, here we have this grating with a periodic pattern that multiplexes our image into 9 images. And now we're going to focus shift them to get that instant 3D series, the instant focal stack. And I had a question break here, but since we were already taking that question, or do we have another one already?
Yeah, I guess the question is the relationship of the 9 images to the stacking of images. How does that?
We're coming to it, right? So the first
All
thing
right.
we needed to do was to multiplex it, and now we're going to focus shift them. So we've taken our one image, we're shooting it out, and we've multiplexed it. And how are we now going to focus shift it? And the answer is with the same optic. So the same optic that multiplexes these images can actually focus shift them. And the way that works is, if we have a fine pattern, then we're going to diffract the light stronger. And if we have a coarse pattern, we're going to diffract the light weaker. So what we do is, here we see an example of that again. If we have this tight pattern, the light is going to diffract more. And if we have, you can think about it, if you're a light wave and you're coming, you're going to squeeze through this aperture or this grating. If the grating is smaller, it affects you more and you ripple more. So there's a lot of math that goes into this, and we calculate that to really reduce, or to refocus the light. So here we have, again, the image coming through the grating. So we have the same angle on top of bottom here. So the beams go out at the same diffraction angle because it's periodic. And now if we shift this, if we put this little chirp into the grating where it's distributed with fine pattern on one side and coarse pattern on the other, all of a sudden these angles are not going to be the same across the surface. And that leads to a focus shift. So the beam here that's coming upward through the grating is going to be focus shifted further away from the sensor. And the beam that goes downward here is going to be focusing closer. And if we do all our math right, and we calculate the out of focus phase shift of each focal plane that we want to capture, we get the refocusing function. So here we have multiple focal planes in the sample, coming through the imaging system, the objective, going through the diffractive optics, multiplexing out, and each image has a different focal plane. So that is the principle of this imaging technique. And here we end up, we can see the distorted pattern. It has that grading function that we designed to distribute light evenly into these 9 images. And because of the distortion, we get that focus shift. So what this means is that I put on my camera and I take a picture, and I have already that whole 3D focal stack. So now I can just push play and start imaging, and the whole 3D scene is being imaged as a time series onto my camera. So here's what it looks like in real life. We have built this imaging system onto a regular microscope, And this is in the biological laboratory of Corey Bargman at the Rockefeller University. She's still a professor there, but she's also heading the Chan Zuckerberg Institute BioHub in San Francisco, where there is a private research initiative by Chan and Zuckerberg to study biology. But this is in her lab on Manhattan. And we have microscopes, we have microfluidic systems to put specimens on these microscopes. And then we have this mysterious black box. And inside the black box are the diffractive optics. So you can see these different lens holders in array, and they contain lenses to relay the optics and then the diffractive grating that multiplexes the beam and forms this instant focal series. So here's an example of live data from that microscope. So these are centromeres in yeast. And this is what raw data looks like. A lot of people don't show this because it looks kind of not so clean, but I figured it was good to show what the actual raw data coming out of this imaging system would look like. So we see these little dots that are the centromeres, they're fluorescently labeled. We see a little outline of the yeast cell, which is splitting. It's a Saccharomyces yeast, so it's a fission yeast, so it divides in two. And we can follow the centromere in both of the sister cells. And as you can see, if you look at only one image plane here, that little guy is going to go out of focus. But if you see all of these image planes, and you get used to it after using it a little bit, and I realize it's not the easiest thing to see the first time you look at it, so therefore I've made this this 3D visualization. And now we see them green, which they really are, and that camera records green light. And we can see them diffusing throughout this 3D volume of the yeast cell, and we can track them. So that was the goal of this imaging system, right? So this was the reason that we split the image up and refocused each plane, because now we have this 3D view of the specimen and it's simultaneous. We don't have to focus through the samples super fast to try to follow these two little guys diffusing through the volume. And here's another example. We have a cell nucleus. We have RNA polymerase in the nucleus of a cell. And these are single molecules. So they have very little fluorescence signal, and they're also moving fast. So this is very challenging imaging. We have another example here where we have mRNA diffusing. And like I said, it's really hard to visualize 3D data without these 3D goggles. So what I've done here is I've put a little rotating sample up, and hopefully that's easier to see the volume of. So you see here in red, the nuclear pore complex. You see in blue, the chromatin, which is stained. And then on the right, on top of that, we've visualized the mRNA in that very same cell. And it's diffusing through the nucleus. And what the biological researchers are trying to see here is, how does mRNA get transported inside our cell to find chromatin regions to start transcription of DNA? It's really exciting research. And the hope is that this type of imaging, where you can actually see the cell, you can see the 3D landscape of the nucleus, you can label different things inside it, and then same time track the mRNA diffusing through it, that we can figure out how this works. So do we have more questions? Is it now clear why we did all this diffractive optics?
Yes, I think that is clear. But we do have some more questions. Let's see. Does each photon only end up in one of the images?
Yes. Yes. So we need multiple photons, which means that we're splitting light into 9. So when we do the focus stack, we have the same situation, except we can capture all of them now, right? But each photon can only go to one place. So we're going to have more light when we do this. And this is one of the challenges in 3D imaging in general. It is not different from the challenge in the focal stack, because there you record 9 images. So if you record 9 images with the same exposure time as this image, you're not going to have enough signal. And this is why the work of colleagues of mine who are developing fluorescent proteins is so important, because the goal there is to develop proteins that can emit a lot of photons without bleaching. So there's always, like, microscopy, when we teach, we say that microscopy is an unfortunate series of compromises. So whatever you want, you have to pay for it. If you want signal, you have to pay for it with laser illumination, and you have to get that fluorescent protein to shine. If you want a 3D image instead of a 2D image, you need more light to record that image. If you want to image faster, you have less photons per time frame. So then you're also paying for it by having less signal. And this is why the work that's going on right now with certain groups like Loic Royer and Florian Hugh, who are trying to de-noise data, because what our problem is really noise, right? So we have signal to noise. If there was no noise, we could see everything. Even if we have a really weak signal, if there's no noise, we can see it. So they are trying to use a lot of deep learning methods in order to really reduce that noise and allows the image with less light, so that we can go both faster and in 3D.
Okay. Does the diffraction grating give you super resolution by itself, or is there a pinhole disk involved as well?
So the pinhole, I think, is a reference to confocal microscopy, which is a smart thought, because what we're going to talk about this a little bit later. So no, this is not super resolution. It is maintaining the full resolution of the microscope, but the super resolution we have to add on in a different way, and I will get to that also. And we do actually use diffraction to generate that. But the pinhole is a reference to confocal microscopy, and that is when you scan the sample, and that is much slower. But we will get to that. It's a great question.
Okay.
Yeah.
You might get to this one as well. Clearly, this is a person who is knowledgeable in the area. So
Yeah.
what if you add a phase change to the grading? Would that give you yet another degree of freedom with which to work?
So that's what we do, right? We do a phase shift. So the phase shift of the wavefront is the focus shift. The grading is a phase grading. So the stair step there, they introduce a phase shift to the light. And you could, well, now I don't know if this what this person meant, but you could also try to do aberration correction of sample aberrations in top of the optics using distortion of this, but that would be an adaptive optics application, and you wouldn't be able to do it with a glass grading. You would do it with a spatial light modulator. And we hope to do that in the future, but at this point, the spatial light modulators are not photon efficient enough. But they are really exciting technology. And then you could add, this distorted pattern, you could distort it in different ways to compensate, like we do in atmospheric physics when we do astronomy and we have adaptive optics. If we're looking through the cell and something in the cell acts as a lens that gives us an aberration, and we could compensate for that. But that's something that this person and I should really discuss over a cup of coffee on the wharf in Santa Cruz someday.
Last second, or just last question for this section. Through these techniques, you are working with scientists who are using the optics, of course, and using the microscopes. Have they discovered anything that you could relay that's unexpected? I mean, they've kind of observed some things, but what have they learned?
Yeah,
something they've learned?
Yeah, I think the transcription imaging of what I've shown so far is the most exciting, that it's becoming possible to follow these molecules in 3D. It is really hard, and it'll take years before there are actual breakthroughs. Robert Chijan, who's at Berkeley, did some beautiful imaging with this, looking at these polymerases and RNAs, and there's a Cell publication on that. I will not try to explain that work because I do not understand the biology, and it's shameful. But yeah, this imaging is for basic research in biology, and that is the kind of questions. And I'll talk a little bit more later about applications.
Great, thank you.
All right. So the application is apart from the first thing we did was looking inside the cell nucleus, right? And then I came to Corey Bargman's laboratory at the Rockefeller University. The reason for that was actually a great tragedy. And Corey took me in when my PhD advisor got a brain tumor, and she rescued me and gave her space in her lab. And in her lab, they studied neurobiology and tried to figure out how our brains work. Now, people mentioned fMRI and those types of methods of imaging that we can do on humans, right? But Saralab takes the approach of saying, let's look at a simpler system. Let's look at a model organism. So there are these transparently worms that anyone who ate an apple or something today probably ate one of these little guys. They're not dangerous to us because they eat vegetables. They're about 1 millimeter size. They're transparent. And in them, we can express these fluorescent proteins that we discussed earlier in neurons. So here I'm showing a C. elegans worm, and then I'm showing him with his nose pointing left inside the microscope. And the little green blobs that you see are neurons in this worm's brain. And they're labeled with the green fluorescent protein so that they glow in the dark. But this green fluorescent protein has been modified with calmodulin so that it actually only lights up when the neuron is active, which means that we can visualize in real time in these living animals neuronal signaling. So these particular neurons, and the reason I chose this example is we all know that there's a pandemic going on and people lose the sense of smell sometimes when they have this infection, and these happen to be olfactory input neurons. So these are the worm's smell neurons, and that's an example of how can it be useful to look at a worm. Well, the worm also has smell. We want to understand how this nervous system works, how these neurons work together, how they integrate to the rest of the brain. And what the worm does, it smells this fruit and it goes to them and it wants to eat them. So here's the worm. And when I came to the lab, the students in the lab, because it's students who work always, and one should always be clear that, who does this work? It's PhD students, it's undergraduate students, it's postdocs. Like these are the people who drive the research. This is the locomotive, the driving of this research train. So what the students were doing in the lab was they're looking at 2 or maybe 3 neurons at the same time and trying to figure out their interactions, because they were, again, limited by this 2D imaging problem. So what we did here, so here's the worm, and it's lying in the microfluidic chip, and it's sticking its little nose out, and we can flow smell past it. So we're flowing smell past its nose, and then they're looking at 2 neurons signaling and trying to figure out, what is this neuron doing? Is it telling me that the smell smells good? Is it telling me that the smell is dangerous and that I should stay away from it? So we put this multifocus microscopy technique to work on these worm brains. And here are these sensory input neurons from the nose signaling in the living animal lying inside the fluorescent chamber. And now we're able to follow the whole set of sensory input neurons. And I think it's absolutely beautiful. What we're seeing here is that 3D image sliced up into those 9 planes. And there's actually mirror neurons. If you look at the top and the bottom, they look similar. It's not the same set, but there's one set on each side of these neurons. And it turns out they actually don't fire at the same time. That would be just redundancy. And we're trying to follow that here, so we can see the neurons going on and off when the neuron is active. And what's happening during this recording is we're flowing in the smell of almonds, and then we're stopping the smell of almonds. And in order to make sense of this data, we teamed up with Daniel Soudry in the Paninsky lab at Columbia University, which was on the other side of Manhattan. They had some beautiful data analysis methods where they could, instead of having a graduate student sitting and clicking on the neuron, drawing a region of interest and plotting the intensity profile from the data set, we could automate this. And automated data analysis is something super important. And there's great efforts at UC Santa Cruz, both in data storage, data acquisition, and data processing. We have leading people like Dara Long running these data centers and data server centers. Imagine how much data we are generating here and how we're going to look at it. So here we see the different neurons. So we have a set here of the smell coming in, and the pink little band indicates when the smell is on. And we see that some neurons turn on. So the neuron called ASI turns on when the smell goes on. But there's also a neuron that turns off when the smell comes on. And again, this is not my field, so I have not sat and analyzed and figured out how these neurons interact with each other to cause behavior. But the researchers in Corey's lab and in Liam's lab are working on this type of problems. So this was really cool. However, the biologist pointed out to me that 9 focal planes of a focal stack is really not that much. I mean, it was great that we could image these 2 sets of sensory neurons, but if you look at the brain of the worm, there's so many more neurons. So they were telling me, like, I mean, sure, Sarah, this was great, but like, I would like to actually image this whole brain imaging. That's what I want. And this is just a worm, right? I mean, can't you do that? And we thought, okay, we're going to need a bigger microscope. So we thought, okay, let's do that. Let's try to figure out a way to make this bigger volumes. And it turned out that this work ended up being done at the Marine Biological Laboratory where JAWS was recorded, where they needed a bigger boat. So my student Ed, he was visiting biology collaborators at in Jennifer Morgan's lab, who are looking at spinal cord regeneration. And the first step in order to make this bigger microscope with more focal planes was to redesign the grading. So we took the picoscale flipper and we said we want a target of 25 spots. So remember, we have the 25 spots is going to now create 25 images instead. So we run it through the algorithm and we find a new grading function, which we see here. And so we make new gradings with this function in it. And we have this design here. And it turned out that the reason, so I actually thought of this idea a long time ago, but then we tried it, but it was really not good because the sensor of the camera is limited, right? So if you want to now take 25 images into that camera, that was not practical. We didn't have enough sampling for that. But luckily, technology moves forward. And the frustrating thing when you're inventing new things is like, if there was just a device, you know, that was a little bit better than what we have today, we could do this. But if you wait a few years, those devices will come. And what came was these tiny cameras. So we were able to actually afford to buy 25 of these, put them into an array, and have one image on each camera instead of us before putting all the images on one camera chip. So the new design looks like this. We have the microscope, we have the diffractive grading with a new grading function that sends out 25 images instead of the 9. And here's the first image we took with that microscope. This is dye-filled neurons and dendrites in the lamprey spinal cord. There's a wonderful philanthropist at Cape Cod who donates money to spinal cord research. She is in a wheelchair herself. And Jennifer Morgan and Gal Haspel, who we work with there, they're looking at locomotion. And since we can't chop people's spinal cords off and try to repair them, they do this in a little fish instead. And they're looking at this fish because this fish has the ability to repair its spinal cord. Because nerves grow back, they just grow back really slowly. And trying to figure out both the plasticity of the system that governs locomotion and what enzymes and what substances make nerves grow all of a sudden, and tell them that actually you should start growing now and reform is really important in therapy for these type of damages and traumatic damages. So this was just a test image, and we can see dendrites and neurons in different focal planes. And the first movie we made was of C. elegans again. So that was that nematode that in the previous images we labeled individual neurons in the brain, but now we've labeled entire muscles in the body. So when the green turns greener here, that means that that muscle is active, and we're looking at the worm crawling, and we can try to figure out how this locomotion works, how it's controlled. And my lab provides the optical technology for that. So I'm not going to be the person who solves that neural interaction problem. I just will try to provide the tools that makes it possible to study them. And this is the importance of basic research. Basic research is sometimes, why would you want to do this? Why would you want to study such a simple system? But very, very important insights come from that. And in my lab, what we do is we're trying to advance optics so that biologists can advance biology, so that medicine can advance to the benefit of mankind. And this system that we built at Woods Hole, we're currently launching in the UC Santa Cruz Life Science Microscopy Center. This is a space that was controlled by the Institute of the Biology of Stem Cells, and then be super great of them to let us host this microscope there. And we're so excited to be able to build it and launch it. And we have a happy user here, Dr. Hilansky now. When this picture was taken, he was not a doctor yet. He was one of the PhD students in the laboratory of my collaborator, Dr. Heinrich Berger. And we can see that this one has even more optics than the other one. And that's because we have two color channels. So we split the two colors up. One goes forward and one goes to the side. And we can do simultaneous imaging in two colors. So that was fun. And what we're going to do next? Well, that's the super resolution. So there was already a question about that in the beginning of the talk, and now we finally get to it. So the diffraction limit really limits what we can see with visible light. So in order to see smaller things within this diffraction limit of resolution, we've long used electrons, because electrons have really short wavelengths, and we can do Ångström resolution. We can see if we have a lipid membrane inside a cell, you can see the two sides of the lipid membrane that envelopes the cell. It's just incredible resolution, but the sample is very, very dead when we do this. So here's an example of synaptonemal complex in mice, myocyte, and it's meiosis. So we see the two sister strands of chromatin wrapping around each other, and we can really look at that in incredible detail, but this sample is dead. So this is why we want to do this with visible light. So like I mentioned before, 2014, the Nobel Prize went to super resolution microscopy. There was many different flavors of it, actually not including this one, which is called structured illumination, which was one of the first methods developed by my PhD advisor at UCSF. And the idea here is spatial frequency mixing. And I'm not going to try to go through the whole theory of how this works, but imagine that you have a sample that you can't see that has this fine structure in it, and you put light on that has a different structure, and those two frequencies mix. So like when you see moiré fringes, you can tease out the information under the stripes by taking multiple images with stripes that you slide across the sample in order to find these underlying frequencies. And it turns out that we can get down here in this image, we have 100 nanometer resolution with doubled resolution of the microscope, and we can see the two sister strands of the chromatids wrapping around each other, separated by 100 nanometers. This is absolutely amazing. I was a PhD student when this was invented, and I was always looking forward to combining this technique with multifocus microscopy so that we would be able to do this live. So the structural illumination microscopy technique improves both resolution and contrast. So it's really quite spectacular. It also has this wonderful property of three-dimensionality. So a lot of the time when you do super resolution, you only do it in 2 dimensions, but with SIM, you can really do it in 3 dimensions and also improve contrast. So we're back at the movie with the color-coded depth, because it's one of my favorite movies. There's only one thing with this movie that was problematic at the time. This was taken 10 years ago, and it took 20 seconds to record each frame because of these issues with the focus stack that we have to record in the 3D image, and because we were not as fast with our electronics to generate the structural illumination function. So it's absolutely beautiful, and what we're really trying to do here is to speed this up. And of course, the way to do that is to include it in our multifocus setup. So we put on our structural illumination into this multifocus system, and it turns out that it becomes even more complicated because the whole data analysis and the image restoration becomes much more complex when you do it in this way. But thanks to my incredible mentors and collaborators in the biology department. We have now, when I arrived at UC Santa Cruz, basically they grabbed me when I came in as a completely clueless assistant professor and sat me down on a chair and told me, now you're going to build this microscope and we're going to help you write your first grant proposal so that you can get money to build this for us. And we got the money from the National Science Foundations, and I'm so grateful. This is what funds my students right now and my optics lab and my work. And when we're at the point where actually 2 days ago, my student texted me and said, look, the data looks good now. So he even sent me the video because he was so excited on his cell phone, like, here, look, it's working. So we're at the point now where we want to launch this. We want to start putting biological samples on it. We want to start doing the imaging. And it's kind of funny. So we have here Grant Hartzog on the surfboard, and we have Nidhi Bala, and then we have Bill Sullivan. And I think he's actually giving the Kraw Lecture next time. So this, unfortunately, he has not been able to use this microscope yet because it just started working 2 days ago. But his work in biology will, I'm sure, be very, very inspiring. He's a wonderful scientist and a wonderful mentor. And yeah, I can't wait to see what we are going to be able to visualize with this imaging system. And I mentioned before that the people who do all of this work are the students. So these are some of the undergraduate, and graduate students in my lab. One of the images taken this morning in the lab of Eduardo, who's standing here with his 25 camera array and doing some little adjustments to the optics of it so that we can get those perfect images. Students also have fun. So, we do hiking trips and skiing trips to be able to stand all these long days in the lab, which is often dark because it's microscopy. And I'm really glad we were able to share this with you today. And I hope that you all are with us, being excited about basic research in optics. And if you don't think it's exciting enough with just optics, think about how it serves the basic research in biology and biomedicine. And with that, I wanted to thank you again to the hosts of the Kraw Lecture series. And if you want to download the pixel flipper algorithm and generate the grading function, you can look at the Saralab site and find the software there.
Great. Thank you so much, Sarah. That was great. Very, very inspiring. And, this is the science is something that moves very slowly, and it's painstaking work, and people, who are not involved in it, of course, don't see that. All of the groundwork that's laid to develop these kinds of instruments that allow scientists to then make hypotheses about the processes that underlie life, and then to turn that eventually into something which can be used to fight illness and disease. So it's a very, very involved and long process that requires sustained support, frankly, and the labor of many, many good people. So thank you for your contributions to that. So we have a few more questions here that I would like to pass along to you. One of them is about the relationship of this 3D imaging technique, that 3D, comparison to other 3D imaging techniques that make use of fluorescence. Is your technique require more or less, or is there some advantage with respect to the fluorescence, level of fluorescence?
So like we said, we split the image up. So I'm guessing the question is about photon budget. So we split the image up into 9 or 25, which means that we have to have 9 times more light. But if we take the focal stack, we need to take 9 images. So that part of it adds up. What we always have is a little bit of loss in the optics. And that's why we're going through those extreme lengths of designing and nanofabricating. And my estimate is that when we put this relay optic system onto the microscope, that black box that contains the lens, the diffractive grating, and another lens, we probably lose half of our light. That sounds like a lot, but it's the best. We have tuned everything into total efficiency. It's still heartbreaking, right? Every photon is sacred. You want every photon. So my guess is that we lose half of the light. But when it comes to the fact that we split the image up, it's the same effect when you go through focus. Unless you're doing localization microscopy, then you are actually better off with multifocus microscopy, because that one photon that went to one focal plane would have been lost otherwise, and now we capture it.
Okay. Back to the yeast slides for a second.
Oh, no.
What actually are the two red dots?
The centromeres were green. Yeah, that is that little complex that sends out the microtubules that pull on the chromosomes at cell division. I hope. If not, my biologist collaborators are going to tease me to death. So the centromere, there are these two little tubes, right? And then all of the microtubules come out of them. And when the chromosomes split, they grab one each and pull them apart. I'm almost sure that this is what happens. So like I said, the biology is not my strong suit here, and I hand that over to my collaborators. So ask Bill next Kraw lecture, please.
Ah, okay, that's good.
That's what I'm gonna say.
And can you talk a little bit more about the multiple lights? So multiple wavelengths of light. So how does that, I mean?
Yeah, so the idea here is that we want to be able to image multiple proteins at the same time. So if I want to look at the nuclear pore, and I want to look at DNA, and I want to look at mitochondria, and I want to look at endoplasmic reticulum, because we have all these different colors of light, we can label them with different fluorophores in different colors. So there's entire palettes today of yellow, green, cyan, blue, and these come from, the proteins themselves come from bioluminescent organisms in the ocean. So there's starfish, coral, jellyfish that have these proteins, and that's where they originally came from. And we have entire laboratories sitting designing these fluorophores that we label our specimens with. And the idea here then is that, you know, when we do this light imaging, one way to do it would be on a color camera. But if we use a color camera, then we lose light, because we're only imaging, each pixel only takes one color. So we split the light with what's called the dichroic beam splitter. It should be called polychroic, because we can split. So we take the blue light on one camera, the red light on one camera, the green light on one camera, and we run them at the same time. So we don't only get simultaneous 3D imaging, we get simultaneous multicolor imaging without losing photons.
I see.
And I know people who have made systems with 6 colors. I've only made 3 myself.
So if you were to think about what the ultimate microscope might look like or be, I mean, you know, you went from 9 to 25, and, you know, is there some sort of end goal that would get you to some place that you could, you know, identify? Or is it just
there's no end. There's no end
smaller,
like to where we can get. But one of the most exciting things right now, I think, is phase imaging and polarization imaging, because these fluorophores are great, but they're finite, right? We only have so many photons from them, and we have to label the specimen is invasive. If I wanted to image my body, I wouldn't want to put GFP in it. But label-free imaging, which was done in the 40s by fantastic, like a Japanese-American researcher, Shinwa Inoue at Woods Hole, he imaged with just polarization and phase. And it turns out our DNA is birefringent. So we can use different contrast methods in label-free imaging. And that, I think, would be fantastic. My student, Eduardo, was at the BioHub in San Francisco, the Chan Zuckerberg BioHub, doing an internship with Charlene Mehta there, where they were looking at polarization and phase imaging. And we can join that with our 3D imaging technology to have less invasive imaging. less invasive, I think, is key.
Yeah. Let's see. What is the size of the image that you produce? I guess measured in pixels.
Well, I would say measured in pixels and in the sample too. So the field of view of the 9-plane microscope was 33 microns in the cell. And that, back in the days, we put on a 512 by 512 CCD camera. So the field of view of the neural images with the 25 planes, that was 50 micrometers. This is all very small. And the focus step between the planes was different too, because you tune that with the grading. So if you want an image with really high resolution inside the cell nucleus, you want those slices, you want to slice them up in like maybe 200 nanometer slices, maybe 100 nanometer slices. And when you're looking at the whole brain of the C. elegans, the neurons are giant. They're 4 micrometers. Okay, it's really small, but it's bigger than the cell nucleus. So then you can space your focal planes by a couple of micrometers and you get a much deeper volume. So everything is tuned to the sample. So we work very closely. I was actually asking Needy, my collaborator, in the corridor this morning, hey, which colors are you going to use? Because we want to know what color, and then we want to know what step. So everything is just, it's really a custom, it's bespoke microscopy design for the sample.
Yeah. Have you looked at the metal lens technology as a way to generate the structured illumination? I do not understand what the question is asking, so one thing you could do is explain the question.
Yeah, so structural illumination microscopy, the idea is here to generate that pattern. So we're putting these stripes on, and I call them stripes, but it's actually very simplified to call them stripes because they're only stripes in 2 dimensions. In 3 dimensions, it's like little eggs in space. Like if you put 2 big egg crates on top of each other, we have little eggs of light traveling through the sample. And the way to generate that is really, really challenging. You have to basically, what you're doing is you're building an interferometer inside your sample. So the way we do it, in the first imaging experiments, we used diffractive gratings to form these patterns. What we do now is we use what's called a spatial light modulator because it's tunable. And we bounce the light off that spatial light modulator and we shine it into the sample. I'm sure there are many more ways to generate the pattern. For now, we are super happy with the spatial light modulators because they're so fast. We can generate the whole sim series 25 times per second. It's really good.
Where does the image actually go? I mean, is it, does it go through a microscope or is it?
Yes. So here's the microscope, right? So here's Robert, and he has his microscope. And you see there, he has an eyepiece that he looks in when he's going to find his sample. And then he switches the light. And normally it would go to a camera on the side of the microscope. So now we took that camera off. And we built this black box that you can see into. Normally there would be a lid on that because we don't want that optics exposed to anyone. And it's frankly intimidating to look at too. So we just want to hide it there. And it's like the black box does it. And then at the end of the black box, that's where we put the camera back on. So that's where the image comes out. So you can modify, like my dream is that someone takes this and commercializes it. And Zeiss Microsystems in Jena, East Germany actually offered to do so on the condition that I quit my faculty job, leave my students in academia and move to Germany and work for them, and they would commercialize this technology and sell the black box to people. And I couldn't do it. Like I live to be like in an academic environment where we just do research for the sake of advancing knowledge.
Well, let's close with a question actually about the students. You have this penchant for having undergraduate students in your lab. What kind of skills do they come with, and what sort of how do you choose the students who come to you? How does that work?
You know, the students come to you and they say, I'm really interested in your work, and then they already have you. If they come to you and say, I'm so interested in optics, I love microscopy, you know, and then they say, I have this idea, I would like to do this project, or so students came to me this year saying, I want to do deep learning on your data.
Oh,
And I said, thank you, please do deep learning on the data, come straight into my lab, look at our data here. What would you like to do with this? Because these new methods are so powerful. But I think like for pedagogical purposes here, I should point out that hard skills are important. We need to all study mathematics and physics at basic level, and biology. So that's where I now have to go and read up a little more so I can explain better next time what the centromeres are, because we really need to understand each other's fields of research to work together. But as a student, building hard skills is a very good start. Mathematics, physics, biology, all of these topics. And then you go and apply it in somebody's labs by getting interested in a project and working on that project and working with other students and learning from each other.
yeah, that's wonderful. Okay, well, thanks again, Sarah, and I will turn it back to George.
Thank you to our panel for sharing this important research with us tonight. Please join us for our next Kraw Lecture, October 20th, Microbes, Big Sur, and Global Health with Professor Bill Sullivan. Good night, everyone.